Welcome to today's clinical overview on molecular orthopedics. While mechanical forces provide stability to a fracture site, a strict molecular blueprint is what actually governs the biological repair process. This essential clinical synthesis was compiled and presented by Assistant Professor Dr. Farhad Mustafa Musa Atrushi, a board-certified orthopedic surgeon. At the center of this lecture is a single cellular choice. How do extracellular protein signals dictate whether a mesenchymal cell divides to bridge a gap or differentiates to produce bone? This diagnostic matrix organizes those signals. You can see from the headers that these growth factors are strictly categorized by their receptor class, which defines their core clinical function. To safely utilize biologics in the OR, a surgeon must be able to decode the rigid split between osteoinduction, which is differentiation, and mitogenesis, or cellular proliferation. We begin with the serine-threonine sulfate receptors. These are targeted by TGF-B and BMP to signal undifferentiated mesenchymal cells to become active osteoblasts. This cutaway reveals the primary mechanism of osteoinduction, the BMP pathway. The process starts at the cell membrane, where a BMP molecule binds to a transmembrane complex consisting of type 1 and type 2 receptors. This binding causes the receptor to phosphorylate at the GS domain. This triggers the activation of primary intracellular mediators called SMATs. Specifically, our SMATs are phosphorylated to form a complex with co-SMATs. This SMAD complex then translocates through the nuclear envelope to enter the nucleus, where it directly transcribes the target genes responsible for bone formation. Beyond this primary axis, BMP signaling can also trigger alternative MAPK pathways, such as ERK, JNK, and P38, to further influence gene expression. This direct, uninterrupted pipeline from the membrane receptor to the DNA is what gives BMP its potent, unavoidable osteoinductive power. The strength of this pathway is demonstrated by its clinical failures. Genetic mutations in the BMP4 gene cause fibrodysplasia ossificans progressiva, a catastrophic condition where soft tissues are forcibly converted into bone. Mesenchymal precursor cells can also be hijacked internally before they ever begin to form bone. This diagram shows the PPAR-gamma nuclear receptor. It acts as an adipogenic switch, binding directly to DNA to force mesenchymal cells toward fat differentiation rather than bone formation. Because osteoinduction is so potent, any error in this molecular code leads to immediate and irreversible tissue pathology. We pivot now to the tyrosine kinase receptors. These are the mitogens, FGF, IGF, and PDGF, responsible for cellular proliferation, chemotaxis, and angiogenesis. IGF-1 is the most abundant growth factor in bone. It drives rapid cellular expansion through both paracrine and endocrine pathways. The GH-IGF-1 system induces linear skeletal growth by driving proliferation in the growth plate without causing maturation. Conversely, the active thyroid hormone metabolite stimulates both proliferation and maturation simultaneously. Note that IGF-2 is even more potent than IGF-1 in driving type 1 collagen production and the synthesis of the cartilage matrix. To complete the pathway, PDGF signals inflammatory cells to migrate to the fracture site, while FGF initiates the early stages of angiogenesis. BMP establishes a cell's identity as an osteoblast, while tyrosine kinase mitogens provide the volume of cells and blood supply to fill the fracture gap. True physiologic repair requires all these molecular factors to fire in a highly choreographed sequence. In Phase 1, PDGF signals macrophage migration to the hematoma. Phase 2 follows immediately, with FGF initiating blood vessel formation in the fracture gap. In Phase 3, the IGF axis drives cellular expansion, leading to the terminal Phase 4, where TGF-B and BMP ignite structural matrix synthesis. Fracture healing is a chronological relay race between inflammatory chemotaxis, angiogenesis, proliferation, and terminal osteoinduction. Understanding these pathways dictates the safe application of commercial biologics in surgical practice. Recombinant human BMP2 is FDA approved for single-level ALIF between L2 and S1 and for acute open tibial shaft fractures stabilized within 14 days of injury. RHBMP7 is indicated as an alternative to autograft in recalcitrant long bone non-unions and compromised revision lumbar fusions. The raw potency of the BMP-SMAD pathway demands absolute respect for patient safety guardrails. Absolute contraindications for RHBMP usage include pregnancy, skeletal immaturity, active infection, tumors, or a known allergy to bovine type 1 collagen. Surgical excellence is entirely downstream of molecular biology. Thank you for attending today's clinical lecture.